Hello, everyone. My name is Elizabeth Abidi. I am part of the Truman Lab, and my project involves investigating the role of ydj1 phosphorylation on the DNA damage response. So, as we all know, proteins are important building blocks of life. And in order for a protein to function properly, it must be folded into its correct shape. HSP70 is an important molecule in folding proteins properly. However, it doesn't really do this process on its own and requires help from other co-chaperone molecules. And one of these molecules is called ydj1 that is very common in human and yeast cells. When our body cells are exposed to harmful conditions such as UV exposure or a lot of chemical exposure, it activates the DNA damage response pathway, which could possibly help stabilize our DNA from the damage. Once this damage has occurred, it will send signals through phosphorylated protein kinases to let our body know that DNA needs repairing. And a key player in this DNA damage response pathway is the R&R complex. And as you can see, these subunits are closely participating in some sort of interaction with ydj1. So throughout our research, we have currently explored the structure of ydj1, and we have found a total of 10 phosphorylated sites, and these located on the zinc finger-like region, highlighted in green, and the dimerization domain highlighted in purple. So based on these current knowledge we've gained on ydj1, we hope to currently explore how ydj1 is phosphorylated and how that phosphorylation impacts the cellular response to DNA damage, specifically through regulating the R&R complex. If we can possibly fine-tune the chaperone function, we could possibly sensitize cancer cells to radiation and other stress-like conditions. So how do we study the effect of ydj1 phosphorylation? Well, we have currently transformed yeast cells by eliminating the expression of ydj1, and we inserted a plasmid that could possibly prevent or mimic phosphorylation for each specific site. Within these past couple of months, we've analyzed 3 sites, which include T160, S161, and T183, through a series of serial dilutions where wild-type ydj1 and ydj1 delete served as our control variables. As you can see from these series of serial dilutions, a majority of the strands had a phenotype very similar to wild-type ydj1. However, you can also note that T183E residue under a heat stressor 37 degrees Celsius lacked growth similar to ydj1 delete. This could possibly suggest that the function of ydj1 has been hampered and hindered in some sort of aspect. So what do these findings suggest for the future, and how do we know its impact for the public, and what does it do for you and me? Well, ultimately, we hope that by screening the rest of these mutations, we can find a way to make cancer cells sensitive to these stress conditions through altering ydj1's interactions with the R&R complex and the DNA damaging response pathway. Thank you so much for listening to my presentation.